This video will explain the different defense mechanisms of the body and function of the immune system. But before we get started, we need to get familiar with a few terms. Antigens are surface markers found on all cells of the body. They can either be self-antigens, meaning they are acceptable to the body, or heteroantigens, meaning they are not accepted by the body. Antibodies, also known as immunoglobulins, are large Y-shaped proteins which bind to the antigens. These proteins mainly serve to identify the pathogens for the big eaters. Pathogens can be bacteria, viruses, fungi, or any microorganism that can potentially cause a disease. Macrophages are large cells that recognize and engulf invading pathogens, destroying them. Innate or nonspecific immunity refers to the immune reaction the body uses no matter what the antigen may be, whereas adaptive or specific immunity fights only a particular antigen. Moving on, we have B and T lymphocytes. B lymphocytes, or B cells, are a type of white blood cells which, when activated, produce a lot of antibodies. B cells can also form memory cells. T lymphocytes, or T cells, play a big role in cellular immunity and killing infected cells. But how do all these work together to help the body fight infection? Well, it all starts with a pathogen. Once it has made it past the first line of defense and enters the body, the second line of defense begins when non-phagocytic white blood cells release histamine, causing the blood vessels to dilate, allowing more fluids to pass through them. As a result, the area becomes swollen and hot. Macrophages will then engulf and destroy the invading bacteria. This is where the third line of defense performs its function. Once the macrophage has engulfed and destroyed the pathogen, pieces of that pathogen, called antigens, will stick out on the surface of the macrophage as a marker for the lymphocytes. The receptor sites on the helper T cells will then bind to these antigens on the surface of the macrophage, and this releases chemical messengers from both cells, causing the T cells to multiply. By doing so, the T cells are destroying infected tissue cells and preventing the pathogen from replicating. The B cells will also bind to the antigens to help destroy them. This creates what is called an antibody-antigen complex. The T cells will then bind to this antibody-antigen complex, activating the B cell, causing it to enlarge and divide, producing plasma cells and memory cells. The plasma cells will continue to produce antibodies at a rate of 1,000 per second and release them into the bloodstream. Meanwhile, the memory B cells, along with the antibodies, will remain in the blood in case the pathogen is reintroduced. This is why it is unlikely to get the same infection twice. Your body is now immune to it and can identify it as soon as it is reintroduced.